so much for joining us today. Before we kick this off, I would like to point out to the audience that we welcome questions during today's discussion. Please post them in the Q&A section at the bottom of your screen, and time permitting, we will address those at the conclusion. So I'd like to now turn this over to Stephen Saltstein, Force Family Office's CEO.
Thank you, Callie, and thank you everyone for taking time out of your day and joining us. We greatly appreciate it. We have a fantastic discussion for you around cancer prevention and early detection. And with us today, we have Bernard Peperstraete, the CEO of AcuamarkDX, and the great and renowned real estate developer, Bruce Ratner, who has been a voice in the woods when it comes to early cancer detection. And Bruce, I got to ask you, I did a fair amount of research before today's talk. I watched a bunch of your videos, and the one thing that I concluded that you are is a mensch. You're really a mensch, and I just have to ask, how does such a mensch become such an amazing and successful real estate developer?
That's a good question. I didn't intend to do that originally. I actually worked for the city for New York for 8 years and taught law for 5 years. And then I needed money, so I went in the real estate business, and I got enthusiastic about it. But we ran our real estate company, frankly, a bit different than many others. We were always focused on, we believed in union labor because it made middle-class New Yorkers. We believed in integrating our company. 50% of our management for a development company were women, not because we thought better or worse. It was just a question of who was the best. So we ran a different kind of company, and I felt good about that. And that was very important to me. So maybe one thing creates another thing. I don't know. So
do you think your time working for the city made you more successful because maybe you knew how to navigate the mentality?
That's a great question. The answer is yes. First of all, I loved it. When you're young and you're working for the city and you have an ability to change things that you could never imagine at such a young age. So that was very encouraging. And then what happened was running different divisions within the city gave me management experience, which I don't think I necessarily inherited or had. I was more thinking an idea person. But my real love has always been science. I really basically took science courses at college. And so I've always, and math and science was always my thing, and I think that kind of helped too in real estate.
Yeah, I mean, it seems like, I don't know if you had any input in this other than developing it, but if you look at the Barclays Center, I mean, there seems to be a fair amount of math in the architecture of the Barclays Center.
Yes, that's kind of true. Because we were dealing with, just as an example, we're dealing with a relatively small site. And so I actually took home the size of different arenas all over the country. And on the floor, I mapped them out and saw what one fits best because it was so small. So I found one that fit best. Not that we copied exactly, but I knew it was doable to be able to put an arena on that site. I didn't have architects do it. I did it myself. So anyhow, that's what I kind of did. And that was math is important. I think math is very important in real estate. I think math, I hate to say it, math's important for a lot of different things. So it certainly has always helped me. I think it's helped me think better, and I have a great in math too, so that makes me happy. Anyhow, so
that's great. Yeah, good.
By the way, I would say there's a difference between math and arithmetic. So I was particularly good at arithmetic because my father, who was from Eastern Europe, spoke with a heavy accent in the car. He would say, "Blisky, that's 4 and 8 and 6 and 10 and 10," and I would add up things very quickly. So anyhow, that's that.
That's fine. The kids today, they would appreciate that with the whole 6-7 going on.
Yeah, that's true.
So, which I still don't even know what that is, but anyway.
I don't either, so.
Yeah, so I don't think they do. So, look, you're one of New York's preeminent real estate developers. You built the Barclays Center. You built, major real estate projects, and you have this legacy in real estate, but you also have this legacy in cancer detection.
Right.
And so, how did you make the jump from real estate development?
That's a good question. So I had a brother who was a year and a half older than me, and our parents died when we were younger, so we were inseparably close. And he was a very famous human rights lawyer, probably among the top human rights lawyers in the country, maybe world. And so he's a year and a half older than me, and he woke up one morning with metastatic brain cancer, which started somewhere else.
Sorry.
So he had actually, they said that he was going to maybe live for 2 or 3 weeks. And so I'm on the board of Sloan Kettering for about 20 years, and they were able to do something extraordinary. They sequenced, this is 10 years, 8 years ago, 9 years ago. They sequenced his genome, and they came up with a mutation that was rare, but there was a drug for it. And so he took the drug, and it was called cabozantinib, and he lived 8 or 9 months, which is, that we had, and I had a lot of good time with him together. But in the process of that, the chief of medicine, who really helped do the work, said to me, we can determine whether the chemo is working by looking at fragments of DNA to see whether or not the chemo is working based on the fragments of DNA. I said, my goodness gracious. And then he said, I'm on the advisory board of GRAIL. This was 8 years ago. And then we talked about early detection. And that's what got me involved in early detection, and also the recognition that my brother having a metastatic disease wasn't going to live. And that I didn't, and even though I'd been on the board a long time, I didn't realize that. Metastatic cancer obviously has a very low survival rate. And so I began to think about early detection and prevention. And I talked to this chief of medicine about it. Unfortunately, he passed away. And that was when I began, it was 8 years ago, to look into early detection. And I read a lot of articles, and I wrote, I basically wrote up what I had read and understood, and from that created a book out of it. And the book is really about prevention and early detection, and the recognition that it's somewhat ignored. Not a lot of money is put into it by the federal government. And there is something about scientists, and they're great people I've met, and doctors and so on. But Bert Vogelstein, who is a renowned scientist, probably the most important cancer scientist in the world, and he is the most cited scientist in the world, period. But he says that what happens is, if he prevents colorectal cancer, for example, from happening altogether, big deal. If he gets a cure for it, he gets a ticker tape parade. And so I think that's part of the problem. It's not only economic, but it's scientists are, want to advance themselves, and coming up with a cure for a stage 3 or 4 cancer is not very doable, but a big deal. And whereas prevention, you don't get a ticker tape parade.
Right. So. Yeah. Bernard, you run one of the most effective early detection, specifically around colorectal. Can you first tell us the stats in regards to if a cancer is detected early, what the stats are for survival, and then also tell us, what does AcuaMark do?
So maybe I start with the second question first. But first of all, thank you for joining, Bruce. It's an honor being here with you. It's just amazing.
Without doubt.
And I want to just mention to the audience, the book that Bruce wrote, it, had some great accolades, and some people that are very famous, like Siddhartha Mukherjee and Katie Couric had great reviews of it. So you should definitely.
Bruce, what's the title, by the way, before we lose this?
It's early detection. Catching early cancer when it's curable.
When it's curable. So
okay, great.
It's very simple. We discussed the title for a long time. Just decided, let's just say
exactly.
I want to add one more thing. My enthusiasm and my impulse to increase prevention and early detection is intense. It is really what I spent all my time on. It is the most important thing with respect to cancer. And I've been around cancer now for 8 years, a lot. And there's nothing more important than early detection and prevention. I don't believe we're going to have any cure for solid tumors for a long, long time. So,
yeah. I agree. Thank you, Steve. Thank you again, Bruce. Now, AcuaMark is or has been working for some time to develop an approach that, for the first time, we think, with great accuracy, can detect stage 1 cancer from blood. And we wanted to have an approach that's really highly accurate for stage 1, which is the area that we care about, but also one that's highly accessible and more accessible in current technologies. And for that, we had to do some gymnastics. And these gymnastics, translate into research, meant that we had to build new methods and new assays from the ground up that can fit on universally available equipment such as qPCR, which is the workhorse of the diagnostic industry. And many people know the terminology of qPCR from its use in infectious diseases such as COVID, right? And the importance of that stage 1 detection field is survival, right? In colorectal, it's a little different for different cancers, but in colorectal cancer, for instance, stage 1 detection results in 90% survival. Stage 2, roughly in 70%. As of stage 3, which is a stage that we're very good at detecting, the survival is about 50%. If you look across all cancers in aggregate on the globe, current survival rates for cancer are about 50%. And that sort of roughly correlates with a stage detection that is too late, right? And so that's why we so focused on stage 1 detection, which is the area that has been elusive in terms of detection by other companies and other approaches. And it really required us to go through an innovative phase of really reinventing and re-engineering assays that could specifically target ultra-low abundance cancer signal from blood.
So how does it work? How do you use the qPCR and?
Yeah, so it's novel assays, but it's mounted on, universally available, commercially available qPCR equipment. And so the test will go to the lab will run it like a normal qPCR test eventually. And, with the same short turnaround times, just far more accurate signal detection. And the results will go back to the physician who ordered it. In terms of technology, we had to, and this sort of goes a little bit into technical side, but in terms of technology, we had to engineer assays or technology that could, for the first time, do broad panels of, and for the first time, ultra-low abundance methylation detection. Methylations are the markers that we think are the backbone for tests like these. Why? Because you can start seeing things going off track first through epigenomics and methylations. And so we had to really re-engineer qPCR to be able to look at methylations, not just narrow amounts, but broad enough amounts of methylations, and then pick them up at ultra-low abundance that it correlates with stage 1, so that we could get to a test that's ultimately still on a clinical level highly sensitive, so it doesn't have a lot of false negatives, and detects the vast majority of the cases, but at the same time is also highly specific. For instance, a 97% specificity is what we're up to today, and that means there's only 3% of false positives. Why do these numbers matter? Sensitivity and detection rate matters for physicians. I'm a physician. I'm not actually a scientist, but I'm a physician. And so as a physician, I would hate to miss a case, right? And secondly, but as a payer, I would hate to have a false positive. And that's why sensitivity matters for physicians, specificity matters for the payers. And so we developed something that's both sensitive and specific, but is highly accessible, highly scalable, has a healthy margin, has low cost of goods, and remains high accuracy for stage 1 for the first time.
Bruce, you called cancer a solvable tragedy. And by saying that, were you talking about early detection?
Yes, yes. And prevention/early detection really is solvable for cancer. It makes cancer curable. And to Bernard's point, it just isn't curable to any great extent. If it's stage 4 and even stage 3, it's a 50% in the case of colorectal, and for certain things less. And another very important point is catching it in stage 1 is absolutely critical, or stage 2. And there are a lot of other things, because the other thing is, take colorectal, for example. It's no fun having a colonoscopy, and it's not even great fun every year having to do a poop test. That's no fun, and people aren't going to stick with it. I mean, we've had a lot of experience now with trying to get people to come in for lung cancer. Very difficult for someone to come in, take time off from a job, go and get a low-dose CT scan. Same thing for colorectal. I mean, there's been a tremendous amount of advertising and push by the CDC over the years, and they still have only hit about 65 to 70% of the people. And we're missing 30 to 40% of the people that don't get regular colonoscopies or even Cologuards. I mean, Cologuard's a great invention, but you got to do it every year. And then it's only a filtering mechanism. Once you get positive, there's 4 to 5 times it's not going to turn out to be cancer. So that's a lot of work. And that goes to the specificity issue, false positive. So what you've done here, and I want to emphasize something, we have a home office. We do this work. This is very possibly a good investment or not, but we don't invest because of the reason we want to stay neutral when people talk to us about what to do. So I do want to say this, that having a blood test, number one, and two, for something where the alternatives are not things that people like to do is really important. And I looked at your material ahead of time, and your high sensitivity and your high specificity is unusual for stage 1. Just to give you a sense of it, this thing which many of you have heard about, which is GRAIL, which is a great thing. I'm not negative about it, but this average sensitivity, meaning the average stage 1 sensitivity, which means what percentage of the time will you get a hit, is only about 17%. And it's higher for some things and lower for others. So to have sensitivity of the kind of numbers on stage 1 and 2 that you do is unusual and extremely important. It's curable then, in the vast majority of cases. So, looking at it, and again, I don't know all the details of what you've done, although I have a good sense of it. And what you created is something unusual, and I see a lot of stuff. Just enormous number of different things that come across. And you've advanced it. And I will also say your presentation was very good that I read.
Thank you.
I see a lot of those too.
It's a lot of tinkering.
We see a lot of stuff. I see a lot of stuff. And I will say that there are a lot of very good ideas. But when you get down to it's really important to figure out the methodology in which it works. And so lung cancer screening has a problem of getting people to come in and so on. It's really hard. Colorectal, the problem is colonoscopy is no fun. And I'm just repeating myself, and it is not great to have to do a poop test every year. Most people won't do a poop test. And by the way, I have a friend who she calls because she had a case of Cologuard. She tested positive, so she's scared out of her mind. And I say, look, you've got to find out there's a 4 to 5 chance that it's probably not. And it turns out twice it's happened. It's not. She doesn't have colorectal cancer. So that's not fun either. It's a lot of anxiety if your specificity is not high enough. And yours is 97% or something. So that's pretty good.
Yeah, we're at 97%. So it's
Bernard, just so people understand, can you give a sense of how you stack up with the competition?
Yeah, so when we look at, again, the area that we care about, stage 1, stage 2, stage 1, the ceiling, so there is a couple of sides to this, right? So we have a ceiling for, so let's put it this way. There is, let's call it 2 large groups out there, right? So there is the group of the companies that use technology that's called next-gen sequencing, which is a very elegant technology. It's very granular. It is very good at assessing signal that's known to be there, but it's not originally designed to pick up ultra-low abundance signal from a very thick layer of noise, such as what blood presents itself with. And so that's why we see for stage 1, we see roughly a ceiling of 60% for the best performing ones. And then we look at the other side of the equation where we have, companies that have used older existing qPCR-like techniques, and they apply that, for instance, on stool, right? There you see the stool test of for instance, Exact Sciences, the Cologuard Plus test being a very good test. But we still here in the US with 44 million people who are not doing screening. And that has been sticky. That has been sticky for 10, 15 years, as long as that test has been around. And
Why
so
is that, by the way? I mean, are people afraid, or what's going on there? It just seems so simple.
it's so the practicality, and I think Bruce was sort of touching upon it too, but the practicality of it is you have the choice. You do an FIT, number one, you do an FIT test, which is a cheap test. An FIT test, fecal, histochemistry test, looks at the blood in the stool. That's a coin of a that's a flip of a coin. So accuracy is not that great, but it's cheap. You have another choice, which is you can do colonoscopy, which is gold standard recommended. But that takes 2 days out of your schedule. That causes a lot of prep, and you need to recover from it. And let's be honest. I mean, there is a great variance of art and skill involved in the way that's conducted. So there are side effects to it sometimes, and there's perforations that are often not talked about. And then there is the choice of Cologuard. And Cologuard, once you at a GP's office, for instance, have discussed the need to do screening, and you've refused to do colonoscopy, then the GP may say, "Oh well, why don't we prescribe, the Cologuard test, a second step down?" Then you still need to wait for that box to come. You still need to open that box. You still need to deposit the biospecimen in it. You still need to mix it with chemicals and still need to go and ship that box out. It is just not very convenient. And so in terms of, I'm not sure whether that's the problem, but it's just a lot of steps for people to wrap their arms around. These are busy times. And so we think that the versatility of a blood test, right? So a blood test can be taken anywhere. It can be taken at your GP. It's not a new needle. It's a needle next to taking your lipids, right? It's just another tube, right? It can also be taken if you go into specialist levels where many people actually get their testing done because they have chronic diseases or something like that. It's got the level of versatility whereby you can imagine that if a patient visits a specialist, for instance, for, I don't know, checking on their diabetes or so, right? That the specialist checks, well, is this person up to date with their screening? If not, just take a blood test. Okay, send it to the local lab, send it to the hospital lab in the case of our test, right? And so I think it just needs to be seamless and convenient. And if you look at products that make it, they often have both a little bit of a pull. Of course, they always have a bit of a push as well in terms of marketing and appeal, but they need to have a bit of a pull too. And that seamlessness and convenience, we think, is a strong pull factor.
Bruce, a lot of family offices wrestle with balancing philanthropy and return, right?
Right.
How do you think, or how does your family office balance investments like AcuaMark? You're not an investor, but prospectively AcuaMark, that promises both societal impact and potential financial return.
So we just had my family office meeting 2 days ago, and I have two daughters who are older, and they're very progressive in the way they think about the world. And we talked about different investments in private equity, and the issue of health insurance companies came up. And I shook my head. My daughters shook their heads. We don't do oil investments. Now, there's nothing wrong with that, I'm saying, but we're not furious, but we are kind of especially with the younger people, and they're not going to want to invest in oil companies. They're not going to want to invest in things that are unfair in general, making generalities. And so when we invest, we don't want to do stuff that we think is not good, or we certainly don't want to do stuff which we think harms. So, for example, a lot of private equity firms invest in nursing homes. And the only way to make money when you do that is eliminate, nursing homes are primarily, people. And so the only way to get the cost down are reducing people. Well, I certainly wouldn't want my parent or grandparent to go to a place that was owned by a private equity firm that was trying to reduce the number of people. So we like to find investments that are at best neutral, or at worst neutral, but hopefully that do positive kinds of things. And listen, we all live with contradictions, so I don't want to try and say that we're any purer than anybody else. We all do live with contradictions. Look at us, So, so it's very important to my children and to me that the values of what we're investing in are really important. And so I can't emphasize that enough. And so that's why, in a way, even what I do now, even when we do our investing, we use a particular company to do it, and they recommend different things and so on. It's Goldman Sachs, and so they know us very well, and they know what we will do and won't do. And so I think we try and mix both of them, and you can do it. Obviously, it's not that hard. I'm not talking about doing anything necessarily way out, but you can find companies that are anywhere from neutral to positive, and still do very well. So it's important. That's how we invest.
I mean, if you think about AcuaMark, they're very innovative, right? I mean, do you think about when you are putting your stake in the ground, accelerating innovation, where maybe traditional venture capital is not going to play, but like family offices are more qualitative. So
yeah. So the answer is no. We're not terribly venture capital investors, although we have done that. So I'm not sure what your question is exactly.
Well, what I'm saying is family offices can fill the void of accelerating innovation in places where traditional venture capital will not.
Yes, that's correct. Yes, and actually, this example is a good example. You can accelerate something which is really important. Every single one of us has, whether it be relatives, parents, hopefully not, parents, hopefully not siblings, grandparents that have had cancer and have died of cancer. We all know friends, everything. When I speak in different places, I watch the audience's faces, and I see how interested they all are because they all have relatives, friends, maybe themselves that have had cancer. So it's I don't want to say it, I'll use the word, it's very appealing to be able to know that you're contributing to trying to solve something and help people, including your own family, by investing in something. And we rarely get that kind of opportunity. And, it's one of the I deal with so many people who have cancer or have friends with cancer, and helping them get into and part of Sloan Kettering or Weill Cornell, both boards I'm on. And it gives me great satisfaction to do that. And I deal with the families. And it is as all of us know, it is devastating to see a relative friend that has cancer, even if, assuming they even survive. It is hell. It is absolute hell.
Right.
And I deal with it, day in and day out. And so I think I see people wanting to invest, people's interest in prevention and early detection is enormous. By the way, I will say, even the scientists and doctors that I meet at Sloan Kettering, no matter what they do, even if they're not even if they're therapeutics, they all want early detection and prevention. It's really something that they believe in because they know that they've seen the patients and the suffering of a patient who has cancer. And it's almost the anxiety.
Right.
Women with anxiety of breast cancer is enormous, understandably. And,
sure.
and men prostate cancers, of all the cancers, and colorectal. We have a friend right now who has colorectal, and my wife just visited her in North Carolina. She probably won't make it. She got caught too late. So
I'm sorry.
So
Yeah.
Bernard, tell me more about AcuaMark. Where are you guys with the FDA?
Oh, so in terms of our next step, so not necessarily want to spend too much time on the Series B, but the next steps, what we raising a Series B and what we want to accomplish with that Series B is we want to get ready for a PMA pivotal study, which means we need to bring our samples set up to about 3,000 or so. Why? Because we want to do more training with the machine learning and AI models that we have. And then from there onwards, we just do the PMA study, which will need a subsequent round. But at the time that we start a PMA study, we'll have commercial system lock, which means we can start commercializing in a very modest fashion as an LDT, but at the same time also run the pivotal study for the FDA, which will be a large study and a subsequent round, but it will be one whereby we already have a very good idea of what that performance should look like. right? Because we actually don't cut any corners. We want to de-risk as much as possible upfront, which is why we sort of go through the effort of going through pretty sizable cohort of subjects and clinical research subjects before we go to the FDA. So we want to really be sure that we are accurate in what we're proposing to develop through regulatory path. And so the regulatory path, very simply, will be, and investors will have a say in this, of course, but as it stands today, it will be LDT to PMA.
So you guys have a model that seems to be particularly scalable because of the distributed kit model. Can you talk a little bit about that and how that might help you not just be successful in the US, but globally?
Yeah, so one walks always before one runs, right? And so initially we'll have a short period whereby we'll have a small CLIA lab. That small CLIA lab will develop the test and bring the test through the FDA. But very closely trailing that, we'll have the development of a kit, and that kit will consist of a number of plates. This is qPCR technology, so that means we can put much of the technology on the plates themselves. We put these plates in a kit. The kit can be shipped to whichever lab that has the equipment, and this is qPCR equipment, so many labs do have that equipment. And so this means that in future, if one wants to run our test, it can be run in the hospitals. It can be run at LabCorp. We can be a reference lab to a reference lab, and so on and so on. And so it should be universally available. And then in terms of deployment across the globe, well, a kit can be shipped, right? A kit can be shipped. It can be shipped to a lab in Europe, for instance, or in Asia. And the way it works is each kit is slightly differently configured. And so that means that the raw data that comes out of the testing in, say, for instance, that European lab, the raw data comes back to us, we interpret it, and immediately the result is being sent back to the ordering lab. And why is it structured that way? Well, because we're working with small packages of data, right? So each test is a small package of data. It's not terabytes of data. It's a small package of data, but many tests times small packages equal many data. And so we want to keep on training these machine learning models to the extent we can to keep on making it more and more and more precise, even beyond the numbers that we see today.
Bruce, one last question before we go to a few audience questions. I mean, if you were addressing your family office peers, the other principals, what would you say to them to motivate them to step into cancer prevention?
I think a little bit of what I said before, which is that we've all been touched by it. And what I would say to my daughters, they're older, but I would say, look, we've all been touched by it. I lost my brother. You've lost a sister-in-law. And you've seen what they've gone through, the months and months of uncomfortableness, of anxiety, of sickness, and so on. And what an incredible thing that we can do two things. We can both invest, which is important for us, and second, do something that is helpful not only to us, but for many, many people. And I will comment, having a blood test is a tremendous positive. I mean, we're doing a pilot with a hospital here in New York on lung cancer screening. And again, it is very difficult to do lung cancer screening where they have to come in for a low-dose CT scan, and people don't like to go in through hospitals and so on. So having a blood test is actually, we really think that a blood test is the way to go, and probably the only real way to go that you can get large numbers. I just had a blood test. I had my annual CBC test the other day. It was nothing. I walked about 3/4 mile. I went in. I had a 10 o'clock appointment. I was out by 10:05. So I got a good walk for the day.
Yeah.
10,000 steps. There you go.
That's funny. Callie, there's a bunch of audience questions, so I'll let you take it away.
Okay, great. Bernard, how does your assay perform in asymptomatic populations where biomarkers concentrations are low, the lowest?
So the asymptomatic space, I mean, stage 1 is often asymptomatic, stage 2 is often asymptomatic, right? And so we have the numbers of about 88 to 100%, depending on which machine learning model we use for stage 1, 90 to 100% for stage 2, and that's CRC. But if the question is also intended to go, to earlier stage, like stage 0 or cancerous advanced adenomas, for instance, that's an answer we don't know yet. We don't know yet how sensitive this really is and how far back it goes. And that was because, well, we are a poor company. We were a poor company. We've always been very lean. And so we didn't have, with the previous sets of samples, didn't have the luxury to really go and look at the controls to see whether there was any advanced adenomas that we were detecting. Now, I will say this. In our scoring system, so there's, of course, multiple layers when peeling the onion. And so in our scoring system, we have developed confidence scoring. And the confidence scoring tells us how sure are we that a positive is indeed a positive if we see a positive signal. And how sure are we that a negative is indeed a negative if we see a negative signal. And that means we have two sides that we're fairly certain about. But what we did see is in some of the healthies, we saw some fairly high scoring that came out. And it was too high to be nothing. And that means we detected something. We just didn't have the chance to go and look back with colonoscopy what it was. But that's presumably, that might have been things that gave signals such as an advanced adenoma that's turning cancerous, or even a stage 1 that was missed. And so that's important, because catching at that stage, that is the ultimate goal, right? That's the ultimate goal where things are curable and where you can go in for an elective procedure and that's it. So I think those are the numbers, but of course the bells and the whistles, i.e. colonoscopy confirmed controls, is something that we have, of course, implemented in the designs of the studies that lie ahead under the Series B proceeds.
Yeah, I have a feeling that when you're able to do more, you're going to find that advanced polyps, adenomas, and so on will be picked up also. Yeah. I think that's probably true because I look at CBCs at blood counts on different tests and so on, and they pick up slight differences even when there's a polyp, if it's an advanced polyp and if it's a sort of a precancer. So I think that's what you're going to find. But again, I don't know. You would have a better.
Well, to your point, Bruce, our market panels for early stage screening are designed and put together specifically with early stage cancer markers in mind, which cross over into advanced adenomas and polyps. So we've seen these genetic shifts in our earlier years, where we spent several years of bioinformatics and studying this. Yeah.
There's every reason that it would too, frankly. You know, because it's a blood test. So, you know, the blood doesn't know whether it's a polyp that's sending off a methylation signal or whether it's an actual tumor.
Yeah, correct.
And then do you think it's even possible to detect a cancer like pancreatic early enough to save a life?
Well, thank you for that question. I mean, pancreatic is notorious, right? Even at early stages, pancreatic has a low survival rate. So pancreatic is actually, so we have a pipeline. So after CRC, we won't stop, right? So it will, we've seen clusterings in the biological data that allows us to sort of put products together that go broader that go more broadly into certain cancer families. For instance, you can imagine a CRC test won't just stay a CRC test. It will become a GI test, right? Both screening and recurrence monitoring. And GI means CRC plus gastric plus esophageal, right? And that's sort of instinctive, right? It's sort of similar tissue families. And so the pancreatic tests, it's a more difficult cancer, but in the GI version 2.0 type of product that we have identified panels for, and that will only be developed after the CRC test is launched. But you know, that's really GI version 2.0, and at that stage we'll be adding, or we'll be looking at, and hopefully biology will allow us to add, pancreatic and cholangio and liver and so on.
You know, I'm going to make one other comment. Sorry to keep saying this stuff, but so we work with on blood, you know, CBCs, which is combined blood count, which we all take once a year. And what the slightest beginnings of cancer can be seen in your CBC test when AI is applied to it. Meaning, and think of it, you cut yourself and all of a sudden, all of the neutrophils and so go on to try and fight it. So it instantly changes your blood. And so I think you will find, and I know you will, that pancreatic is no different. It takes 10 years for pancreatic to create a tumor. So that's a lot of time that's doing a lot of little things to your blood that before AI you could never measure, but you can measure it now. So again, the ability to use blood is incredibly important. It shows more things than you can imagine. So I think you're onto something very important. So
Bernard, just to follow up on that,
you have to realize something else, which is that even though it's deadly, is rare. So when something's rare, and if you had to do a low dose CT scan of some sort, you'd have to probably screen a couple thousand people to get one positive. And blood allows you not, when you're just doing a blood test anyhow, you can do it without having a person do a million other things. And so I commend you for looking at pancreatic because it's very important. Ovarian is another one that's very important.
Bernard, just to follow up on that, I just want to ask, so how are you using AI? How is AcuaMark using AI in order to get these amazing results?
Well, so if you look at the panel, so we had to really re-engineer qPCR to look at broader marker panels. And of course, these marker panels are not just any markers. We spent some years on sifting through the 5.2 billion or so methylation data points that exists and selecting these panels first for CRC, but we have seen and identified panels for about 20 cancers in total. And so when it comes to, even if it's small amounts of markers, like 40 markers, it's not 10 markers, which is too little and has a ceiling in sensitivity. It's not 1 million markers, which creates your own noise levels if you have to amplify them. It's 40 markers. It's the exact right amount of markers to have a clear picture without creating also your own false positives. So it's that right middle. But so even with a set of 40 markers, you can use help in terms of interpretation. And so, we can use the brains of our scientists, but we can also use neural networks. And so, or both. And so neural networks in our hands works the best. And so it really helps us weigh these markers as in their interactions. Marker A, B, C are elevated. They're elevated together. They elevated to what extent together, and so on and so on. All of these interrelations is something that AI can train, but we're not AI native, right? We are not a company that just wants to solve cancer by just throwing AI at it. We are focusing on enhancing the biological impetus that has now changed with our test. The biological impetus, we're enhancing it with AI interpretation. Eventually, this will just be run via AI, but it's after some tens of thousands of samples.
I mean, AI looks at patterns and complicated patterns. And, that's a huge thing that's changed that allows you to do what you're able to do.
Yeah.
So it's really important.
Yeah.
The human can't do those kind of patterns. That's for sure.
An Excel sheet won't do it. That's right.
Do you envision working with major players like Quest, LabCorp, or hospital systems, or do you plan on remaining a standalone platform?
All of the above. Look, our North Star is very clear. Our North Star is very clear. We want to get this to the patient, and we want to get this accessed by patients as broadly and widely as possible. Business model-wise, it's entirely justifiable to be a test that's available through hospital labs or technology that's available through hospital labs, as well as through LabCorp and Quest, as well as through our own lab, maybe being a reference lab to these labs. We don't know the exact configuration yet, but there are several sort of subgroups in the population that are served by these various structures that currently exist. And so we just want to be available. And so it's sort of all of the above is possible.
Right. And then is a 5-year wait in between colonoscopy tests that are covered by insurance too long? Typically, how long does it take, to go from stage 1 to stage 3? And if so, AcuaMark seems like a much better strategy as it can be included in the patient's annual physical checkup.
Yes, we agree. I mean, the 5-year versus the 10-year is for individuals who have risk factors, for instance, family history or chronic disease like inflammatory bowel diseases, right? That they or their family members suffer from. And so for those, a colonoscopy every 5 years recommended rather than every 10 years. And that's, CRC is one of the cancers. It's part of the reason we started with it. We know it well. It's got high visibility, but also it's a slow developing cancer. And so to move from stage 0 to metastasize, it's 13 years, I believe, the official timeline is. And Bruce, this probably better, but so feel free to correct me, but I think it's about 13 years. So it's slow growing cancer. And so,
we'll finish. Go ahead.
But so the risk factor piece leads me to another thing to say though. And so so if you look at some of the other testing done, stool tests, some of the blood tests, they all exclude individuals that have risk factors. We don't think that's needed for our tests. And those are the individuals that need it most. So to the question of the person in the audience, yes, the strategy is better, although we will not replace necessarily the need to, if our test is positive, to go for an imaging procedure, right? If I were to be flagged by our test, the first thing I would want is imaging, a biopsy, and I would want a full next-gen sequencing profiling for tissue of origin verification, for therapy guidance. And so the future is more like, why should these technologies not all focus on what they do best and then sort of work in reflex fashion and handshake fashion together in sort of ultra rationalizing the screening procedures rather than a bit of a circular standoff between the different techniques that are currently available? Sorry, Bruce, I interrupted you.
No. I was just going to say that it's approximately, say, 13 years. However, that's an average or a median. And the really tricky thing about cancer is certain percentage you just can't tell when they become aggressive all of a sudden. It's really a doubling time. For example, in case of lung cancer, the doubling time can be anywhere from 40 days to 200 days. And you never really know, even though the average might be 13 years, you don't really know whether you have a particularly aggressive cancer, which is really a doubling speed issue. And I have people who I know who went from stage 2 to stage 4 very quickly. And actually, ovarian is probably the worst/best example. Ovarian cancer, you can take a test on day 1, not any good test necessarily, and then you can have a doubling time of 300 days, and then 2 months later it can go down to 40 days, and that's why very often you'll see case of ovarian cancer, someone's fine one day and they're not alive a year later. So the importance of that is this question. Yes, we have a blunt instrument. It's called 10 years and 5 years. We shouldn't have a blunt instrument. We should have a more exacting instrument so that we can take a blood test, because my guess is if you really look at it, and I started actually quickly looking, the doubling times vary. And you just can't. I actually had the other day with someone who had DCI as a type of stage 0 breast cancer, and she has risk factors, family, and other kinds of things, which indicate that there's a 60% chance she's going to get cancer. Now, other people, go from stage 0, which isn't really cancer, to stage 2 or 3 can happen very quickly. So even though the median might be 5 years or 10 years, you can't really always tell, which is why a blood test is really important. So,
thank you, Bruce. Totally agree.
Thank you. And kind of on the heels of that, like, someone had mentioned, Cologuard is methylated biomarkers plus, but yours is blood-based. And how are you better? And what about Shield blood test for colon cancer?
Yeah, so just a brief correction. Cologuard is actually FIT plus some mutational work plus a very small amount of methylation markers, so 2 markers. 5, I believe 5 or so, and then one has 5 variants mutations, plus FIT. So the real backbone of Cologuard is an FIT test enhanced with some genetic work. And so that's number 1. Number 2, and so the Cologuard Plus test, the latest one, not Cologuard, the older Cologuard version, but the Cologuard Plus one has actually very good sensitivity and specificity. It's 94% sensitivity at 95% specificity. In that's being published in the press releases of Exact Sciences, of course. But at face value, that's a decent result. But we just think that a blood-based test that comes within striking distance of that level will be a game changer in terms of changing that compliance, right? Number one. Number two, in terms of the other tests on the sequencing sites, for instance, Shield, again, the stage 1 sensitivities are hitting a maximum at about 60%. And that's sort of in the press releases of the companies as well.
And then how do you study for sensitivity, specificity? You don't have a design lock, so is this just bioinformatics?
No, it's verification on clinical samples and case control studies, right? So the typical progression of clinical research in diagnostics is to start with in silico type of work, and then you move on to clinical work, and that clinical work is gradually increasing cohorts of case controls. And currently we have 350 samples case controlled, double controlled, meaning each case has 2 controls, and we have separate independent training and verification or validation sets. So those are called verification studies. Now that we've gotten to these results, we will benefit in the next study from higher quality samples, colonoscopy confirmed controls as well, before we go and set the design or log the design for the PMA study. Now, in diagnostics and technologies like these, there is various components involved. And so what is locked in our case is the primer pool or the marker pool, number one. Number two is the assay architecture. And the only thing we haven't locked yet and do not want to lock is the training of the machine learning models. Why? Because if we can train it on 3,000 samples, we know it will get more robust before we get to really lock designs for a PMA study. And so this is typical, but the results, even now on these suboptimal conditions, I mean, the samples we collected were not the most optimal samples. We just bought them from vendors, 40% from Latin America, some from Eastern Europe, some from the UK, some from Canada, some from the US. So very diverse, but it was hard to control the quality. And so even with that quality, we had these results. So I think with having some of the top, US hospital systems involved as our next partners, I think that will be a game changer in terms of quality of samples. And just as a segue into that, we just had diligence done by one of the largest integrated development delivery networks or hospital systems in the US for an investment. And so I think they'd be willing to share diligence as well as insights. We had to talk to their heavyweight KOLs in order to get this through with them. And so it came out, it was an incredibly productive and positive conversation and diligence process that we had.
Great. Unfortunately, we are running out of time and have time for one more question. And can you tell us about your capital raise, what you're looking for, use of proceeds? Go into a little more detail on that. Great.
Yeah. So actually, our invention phase is over, and so from here on, it's just blocking and tackling. And so the Series B is really designed to be very concentrated in value creation. And so what that means is actually quite simple. We need to get the technology commercial system locked. Why? Because if we do an FDA study, the FDA wants to know the serial number of the PCR machine one is using. So we need to get that commercial system locked, number one. That's about 18 months of work estimated. We want to bring the sample sets up from 350 to 3,000. Again, to allow us to train the machine learning models even more. And then we want to get the FDA study in the starting blocks, right? So we can start to be in the starting blocks, but having that sample set at 3,000 at that time, at the end of that use of proceeds, will put us around the table with companies such as Exact and such as Natera for MRD and so on and so on. We'll have a small set of samples that will be looking for recurrence monitoring as well. So that moves us into the field of Natera, I guess. And so we're going into the field of Exact and we're going up into the field of Natera. But anyway, so very briefly, system lock, 3,000 samples, PMA start readiness, commercialization readiness. $25 million is the ask.
Okay, thank you very much. And for the audience, sorry we couldn't get to all the questions. Please reach out to me. Everyone has my email address. I'm happy to facilitate an introduction to Bernard, who can answer these for you. And I just want to thank everyone for joining us today, and thank you, Bernard and Bruce and Steve, for such an insightful and great discussion. And I hope everyone has a wonderful day.